I'm Nelly Sana. I'm a postdoctoral researcher here at Hasselt University. I'm going to talk a little bit about black carbon detection in human matrices. So here we're talking about those biomarkers of exposure, the internal dose. So let's talk about this a little bit. Just a reflection about ambient air pollution. There are 4.2 million premature deaths worldwide in 2019 by the cause of ambient air pollution. And this is due to a lot of links with certain diseases such as lung cancer, acute lower respiratory infection, ischemic heart disease, etc. And there are a lot of fractions of these diseases that are caused by ambient air pollution. And 99% of the world's population are living in places exceeding the World Health Organization air quality standards, the guidelines. So actually, at this moment, we are breathing in air pollution. I think it's quite good here in Belgium. We are quite low. It's also raining. That's also a good sign. So we are still okay. Particulate matter is one of the fractions of air pollution, and they poses the greatest health risk. You have those different fractions like PM10, which are like all the particles that are below 10 micrometers, 2.5, the fine particles below 2.5 micrometers, and then the ultrafine particles which are below 0.1 or 100 nanometers. And it are these smaller particles that, of course, can really cross a lot of barriers. Think about the upper airways, like our upper airways, they are more deposited with the 10 micrometer particles, but like going to really the deeper lower areas, the bronchus and the alveoli, we are more at the 2.5 micrometers, and it are really that small particles that can cross the alveoli and then really get in our systemic circulation. And black carbon is one of the fractions that are really compared to these lower particulate matter ultrafine particles. And black carbon, you have already heard about it. It's a fraction that is due to the complete combustion of fossil fuels from cars, etc. So one of the important things, and it has been discussed in the previous lecture as well, is that we are not sitting still at this point. Like if you are looking at external exposure, we are using, for example, monitoring stations to assess air pollution. So here in Belgium, we have, for example, a series of stations, which you can see. Yeah. Which you can see there. They measure on a daily basis the air pollutants in our environment. And then by using models, spatial temporal models, you can assess someone's exposure at the residence of that person, even here in Belgium with a quite high resolution, 100 by 100 meters, but in other countries it can be that the resolution is 4 by 4 kilometers. So here in Belgium we have a quite good resolution to assess the exposure at someone's home. But we are not sitting at home at this point, for example, so you are more dynamic, and then you have, of course, personal air monitors, devices that you can bring along with you, but that's quite expensive. So it is important that we have those internal exposure markers to be able to assess our internal dose. This is, of course, where our story started in 2016 already, so it's already quite some time ago, that we start with the development and application of black carbon assessment in the human body with a label-free way. Because at that point, what could you do is you could label, for example, particles, and then you could ask volunteers to inhale it. and then to see like if this radioactive label was present, but it has, of course, a limited time of duration. So that label-free part is really important. So to start with that, how is this technique developed? It was Hannelore Bouvet that started with this, and she found out that black carbon particles could emit white light when you put them under a microscope with femtosecond pulse laser illumination using a 2-photon laser microscopy. So how is this actually working? And she's on top. You will see several pictures of people popping up, because it is important that it's not my work only, but it's like a teamwork. And what you can see here is actually how it is assessed. So you have, of course, a microscope, a confocal microscope, where the particle is a specimen containing particles is underneath an objective. You have that infrared laser that is in a femtosecond way putting pulses on that specimen. And a femtosecond is 10 to the minus 14 seconds, so it's really, really fast. It goes through a focusing lens on that specific particle, and then that particle suddenly emits white light, which you can detect. One of the hypotheses regarding that white light generation, because we are not sure of it at this point yet, is of the unique energy banding structure of these particles. Normally, you have like straight energy bands, but in black carbon, they are more bent to each other. So you have the valence band and the conduction band. And normally, you have like electrons that can jump to one of the other band, which is then the energy that can be generated, or they can go and transition on the same band itself. And this is what is thought to be happened, that like there is a continuous transition of electrons making an electron cloud, which provides that white light generation. So Hannelore started actually in vitro in cells by exposing carbon black, which is the more pure elemental form of black carbon, to fibroblast cells. And she did a short and a long-term exposure. What you could see nicely is that the morphology of these cells were a little bit changing, but she could also detect them quite nicely within the cells. So the red dots here are a representation of these black carbon particles. Then me and Hannelore did a study. So we collected urine from more than 300 9 to 12-year-old children, just healthy children. And we knew where they lived. We could assess like how far they were living from a major road. And then we assessed the urine, the black carbon. And what we could see is that there was a 9% difference when you lived very close to the road or further away, which was then more than 160 meters away from that major road, in the black carbon that we could detect in the urine itself. So these were like the first results that we could detect it, that it is related to being close to a major road. Within our group, it was important to look at early life health. And then we started to develop and apply the technique on tissue samples of placenta. And why is that? The EnvironAge bird cohort is a bird cohort here in Limburg, Belgium. And since 2010, we are recruiting mother newborn pairs at the delivery ward in a hospital, East Limburg. And since 2010 up until now, we have more than 2,000 mother newborn pairs that are participating. And we have already 2 follow-ups of these children, one at 4 to 6-year-old and one at 9 to 11-year-old. So the eldest children are now 14 years old already, and they can still voluntarily participate to this EnvironAge bird cohort. And this cohort really wants to know like what happens early in life, whether that also influences health later in life, which is, of course, the hypothesis of the developmental origins of health and disease. Of course, when you go to other human matrices than urine, for example, you need to develop other strategies to detect these particles. So for blood and for placental tissue, specific workflows were developed to detect the black carbon particles. So there is another sample processing for these specific matrices. So, for example, for blood, a certain sample is collected between a microscopic slide. Also for the placental tissue, they are like fixed and embedded on a microscopic slide. And how did we test and validate that it worked is by using a spike of carbon black. So we used carbon black particles, we added that to a blood sample or to the tissue, and then assessed actually the concentration of these carbon black particles in blood, and that we knew beforehand, of course. We did it in a dilution series, and the number of detected particles really assessed via the technique. And you could see that there was a nice correlation. Another part is, of course, the white light illumination, that you need to analyze it properly when you're looking at blood samples and tissue, because there is much more in there than in urine. You have cells in there, in tissue, you have collagen and so on. So another analysis technique was applied to assess this. And this was work from Hannelore and Eva. And what they noticed is you have on one hand the 2-photon excited autofluorescence, which is that technique for the white light generation. But you could see also the cells of the tissue, of the placental tissue. So there is some autofluorescence ongoing here. So what they added is another layer of second harmonic generation, which is often done for collagen in tissue. And what they did is they looked for specific thresholds in these both channels. And when they were present in both above a certain threshold, we could be sure that it was a black carbon particle. And then you can get such kind of output data, which is similar to the one of urine. So what they did is they assessed, of course, and they found, of course, the presence of these black carbon particles in placental tissue. You can see that we really need to zoom in and focus on it, because they are very small. And that's normal. You are here in the nanometer range of particles. So we have indicated them with white arrows to be able to show you where they reside, and you can find them at different spots. To really show you that it is really in the cells and within the tissue, you can do a Z-stack of a confocal microscopy. And with a Z-stack, what you do actually is you're going throughout the layers of the tissue in different planes, and then you can get an XYZ image. And what I show you here is a plane just underneath the upper layer. And what you can see nicely is, for example, here a white dot that is nicely within that placental tissue. This really shows and evidence that these particles are within that tissue. How are we sure that we are talking about black carbon? So what we did is we looked at the emission wavelength, which is the fingerprint actually of these particles. And we looked at the autofluorescence signal of the placental tissue, which you can see in green. You can see the black carbon, and you also can see the reference to carbon black. So they have a specific fingerprint that you can recognize to see whether it's black carbon or not. To move forward and to really showcase that it can be crossed to the villus tissue, we used an ex vivo perfusion model, which is actually a model where you externally can expose diesel exhaust particles through an actual placenta for a short time period, up to 6 hours, and then you can assess where these particles reside. And what is interesting is, and that's what I wanted to show you here, is that you could find them in different kinds of cells. You could find them in trophoblast cells, and trophoblast cells are the most common cells that you can find in the placental tissue. You could find them in macrophages, but also endothelial cells like the arteries of the placental tissue itself. But you could find them mostly in trophoblast cells compared to endothelial and the macrophages. Then we are already coming to the 2020 period. And what we then did is to associate these internal black carbon exposures assessed in different kind of matrices here, cord blood, maternal blood, and placental blood, with these external monitoring station data. So we have those data available for all of the EnvironAge participants, and we made correlations to see like how well is our internal dose correlated with the assessment of the exposure measured at residence. And there is a positive correlation. So you can see it's an R of 0.6, 0.5. So it's not a one-on-one comparison, but that's normal because external exposure is not the same as an internal dose. What is interesting, it's a link with residential whole pregnancy exposure. So we used all the data for the whole pregnancy exposure, made an average from this, and this is actually what is associated with this exposure. Then Eva Bongaert also had the opportunity in 4 participants to be able to assess in different fetal tissues. So it were like tissues that were obtained due to pregnancy problems, and then these tissues were collected, and then she assessed whether there is black carbon also really in the fetus itself, and she could find black carbon particles back in fetal liver, lung, brain tissue, and preterm placenta. So it really shows that it goes from mother through the placenta to the fetus already before birth, meaning that we are all already exposed to black carbon even before we are born, actually. That's really important to know, of course, when you want to assess links with certain health effects later in life, especially if development is one of the key processes that contribute to later life health risk. You could also find these black carbon particles in kidney tissues from transplant recipients. And also here it is interesting to show that they are found at different sites within the kidney. Not only like the interstitial region surrounding the tubula, but also in capillaries, in smooth muscle tissue surrounding a blood vessel. So you can find them almost in every kind of cell type. And lastly, I want to show you a small example of black carbon particles that were found in brain regions from Alzheimer's patients. It's post-mortem brain tissue. So what you could see here is that they found these black carbon particles in different regions of the brain. What's interesting is you would expect, because one of the potential ways that we can inhale black carbon particles and reach the brain directly is via our olfactory bulb, olfactory nerve, like by our nose. And you would expect that they would mainly reside and be present in the surrounding tissues in the brain there. But what was interesting here is that you could even find them more deeply. And like, for example, the thalamus, the hippocampus are more the inner regions of the brain. You could even find them more. And that's, with this, of course, we show they are present everywhere. But at this point, we still need to dive more deeply. Like, what does that mean for health? And what, for example, Kenneth van Braben is doing, he is also doing like animal mice studies to really be sure like how and where are these particles residing, when are they residing there, and how is it linked, for example, with cognitive behavior. Upcoming articles. If you want to follow this, please, please do so. So I want to conclude. It is already like we validated the technique for black carbon. It's a label-free method. That's really important. And it's really applicable in different biological matrices and correlated with even ambient black carbon exposure concentration, which also is like a validation of the monitoring stations as well, like how well do they perform in characterizing our internal dose. And you can find them in a lot of different tissues. And currently we are doing a lot of studies because we have, of course, that environment bird cohort with a lot of parameters, biomarkers of effect measured, for example. We do a lot of clinical measurements with these children. So in the future, you will see some articles popping up how these black carbon particles present in the tissue will relate to certain health effects. And with that, I would like to end. So thank you.